Jessica?
He would go out like he would go into the water, catch it, bring it back on the land, and then the other dude would like take him over there.
Yeah.
I used to watch that show. I used to watch Nat Geo, Science Channel, History Channel. American. I remember Gator Boys. Gator Boys. That was Gator Boys. Yeah.
So, we started the endocrine system. This picture shows you kind of an overview smattering of the endocrine organs that we'll be talking about during this section of the course. The first part of the class for our students, as I said, new class is like it's like everybody wants to talk about hormones. You just want to talk, show me the gland and the hormone and tell me what the hormone does. Okay, we're going to do that. But the first thing we're going to do is like the part that students don't like. But it's arguably more important. Like once you learn what the hormones do, it's like, okay, well, everybody knows that. The real question is how do they do what they do? And that's the question that we're really addressing with this. It is the mechanism of action of hormones. So remember, in class last time, we're thinking about hormones in two big categories, lipid soluble and water soluble. And then I gave you some brief definitions of those we were talking about. All right, so here's the additional parts about this lipid soluble, water soluble story. All right, if I say lipid soluble hormone, the hormones that should come to your mind are steroids that are produced by either the gonads or the adrenal cortex. That's good news. There's only two places in the body where we have to talk about these things. The gonads, so either the testes in male or ovaries in females, or the adrenal cortex. There are going to be three families of these in the adrenal cortex that we'll talk about. But the way those lipids work, the way they do what they do, as I said in class last time, is they have an impact directly on the DNA. So the outcome of the binding of a steroid or a lipid-like hormone is the formation of a brand new protein. That's what you get. So we're going to talk about that. I'm going to give you an example so that you can have something to put your teeth into. The other group, the water-soluble hormones, and we have a table coming up and we'll list these, but I'm just going to get you thinking about it. The water-soluble hormones, oh yeah, one more thing on the lipids. You don't have to have a receptor in the cell membrane because they're going right to the DNA. So the receptors for lipid hormones are inside the cell. They don't see the membrane as a barrier, so you don't need to put receptors in the membranes for these hormones. Okay, so the second group, yeah.
You said the receptors are inside the cell?
Inside the cell. They're either in the cytoplasm or in the nucleus. So it makes them like lipid hormones. They don't have any barriers. For example, a lot of things that act on your brain have to get through capillaries in your brain. If they're in circulation, they got to get through to act on your brain. And those are cell membranes. Any kind of lipid thing is going to be non-polar. It's not going to have any trouble crossing the blood-brain barrier. So it makes these hormones really powerful. The only question for the lipid hormone is, are there receptors present? Nothing's going to get in the way of that hormone getting to its receptor. It goes right through the capillary walls, right through the neuron walls, anywhere it wants to go. Okay, the second group are the water solubles, and it's very different. The Water solubles are polar. Most of them are proteins. Almost all of them are made out of amino acids. They carry charge, which means the water soluble hormones, because they're carrying charges, they can't get through the cell membrane. They can't get through it. And so for them to do something, for them to have a mechanism of action, they have to have a receptor in the membrane. So when it comes up to the membrane, the receptor's got to be sticking out because it's not going through. So the location of the receptor is a critical difference. Receptors for these water-soluble amino acid-based hormones are in the membranes of cells all over my body. Now, if they can't get through the cell, then how are they going to do something on the inside? Even if they bind to a receptor, nothing's happened inside the cell. So how are you going to do something, a mechanism? How are you going to do that inside the cell if the hormone can't get in there? So here comes the answer to that question. They have to use what we call second messengers. So they come up to the receptor and they say, hey, I need you to do this. And the receptor reaches inside the cell and passes the message on and says, hey, he needs to do that. And that guy walks to another one and he says, hey, those two guys told me you need to do this. And so it requires other pieces and parts to do its thing. And the beauty of what we've done, us taxpayers, research scientists, pharmacologists, all of these second messenger systems have created these very specific things that we can go after in order to emulate what those hormones are doing in the human body. So as more details become available about these second messenger systems, the better we get at controlling blood pressure and dealing with autoimmune disorders. And I mean, the list is just huge. So I say all that to give you some introduction, but also to try to get you interested in these mechanisms of action. So now my job is to give you some kind of outline with terminology that you can put in place in your head that you can return to. Now, I will tell you, if you were in lab on Monday and you did what I asked you to do, the next 15 minutes is going to be very pleasant for you. If you're in Wednesday's lab, right now you're going to be, I don't know what you're talking about. Try to keep up. In lab today, I'm going to make you go through it in detail. I will not give you all the details in the lecture here that are present in the lab exercise, but I'm going to give you an outline, enough. I want you to have enough terminology so that if you're sitting in a clinic somewhere or your grandpa is taking a drug of some kind, and you type on this thing and you read on this brochure the mechanism of action of what is happening. The terminology you're going to see there is what you're about to hear from me right now. Okay, so you're interested? You want to learn it? Okay, good. Thank you for the nodding. All right. So the first one here is the first one that was discovered. It's the one that we know the most about. It's the cyclic AMP second messenger system. So let me just give you an overview of what we're talking about here. So this is for a water-soluble hormone right here. The terms I'm going to use here are in relationship to water-soluble hormones. And so the one that I used in class last time, I'm going to give you some more details on today, is adrenaline, epinephrine, like the hormone that y'all know what adrenaline does. It elevates heart rate. It increases energy stores, gets free glucose loose in your body. It helps you deal with physically and emotionally stressful situations. Okay, so I'm going to use that one as an example of this. So just imagine this as we're talking about it, and then I'll give you the details. This is epinephrine right here, made by your adrenal gland or by the sympathetic nervous system. And it comes along to the receptor, and the receptor has got to, you see how he's passing the baton? This guy, the receptor, passes the baton here's terminology, to a G protein. The G protein passes the message to some kind of enzyme. In this case, it's going to be adenylate cyclase for this one. And then you're going to get the formation of a second messenger. And this is the cyclic AMP that's going to deliver the message. Okay, so that's a great little baton passing scenario. You need the terms in order to talk about it. All right, so here we go. Number one, so here's your sequence. Number one, the hormone binds to its receptor. It's a water-soluble hormone. The receptor is in the cell membrane. It's not inside the cell, because if it were inside the cell, this hormone couldn't talk to it. The hormone comes along and binds to its receptor. Now, these water-soluble hormones, all of them, the only exception that I know of with this G protein phenomenon right here is the receptor for insulin. That's the only one I know of. Insulin is a protein, and it binds to a receptor. The receptor for insulin is actually a kinase. So in the intervening part here, I'm going to bring that up. But everything else, as far as hormones go in the human body, that are built out of amino acids, that are water soluble, these guys are binding to a protein that is the hormone, and it will be associated with what is called a G protein. Now, G proteins are famous because of that, and these G proteins do a whole bunch of stuff. So right here, in this block right here, there's like a whole world to be talked about. Most of them are 3 subunits. It depends on which subunit comes to attach. The whole thing here. But for you, the hormone binds to the receptor. The receptor then, when the hormone binds, activates a G protein. The first step in water-soluble binding after the binding to this receptor is the activation of the G protein. Now, what that activation means is, I think, too far for us to do. Okay, so we're just going to leave it right there. The next step, after the G protein is activated, the G protein will act on an enzyme. Now, the first one that was discovered was an enzyme called adenylate cyclase. Adenylate cyclase, as you can see, is also in the cell membrane, so the G protein activates an enzyme that's in the membrane. Now, for water-soluble hormones, this is not the only enzyme that is capable of being activated. In the laboratory, we're going to learn about at least one other one, maybe two, I forget. But there are other ones here. So another one is called phospholipase C, and there are others that use different second messengers. I'm not going to take you through all that in lecture. I want you to learn the adenylate cyclase pathway. All right? So step one, the hormone binds to the receptor. Step two, it activates the G protein. Step three, the G protein activates an enzyme. In this case, the enzyme is adenylate cyclase. Okay, I understand that you don't have any real world kind of context for these words yet. So just hold with me, and I'll give you a real world example coming up. Now, when the adenylate cyclase is activated, when it goes live, it's going to convert energy, ATP, into a second messenger. And this is a cyclical molecule here called cyclic AMP. So two of the phosphate groups get chopped off and forms a little ring. Cyclic AMP is the most famous of all of the second messengers in the human body. It is the one we know the most about. It was the first discovered. And here's what we know about cyclic AMP. It activates protein kinases. Now, let's talk about this word kinase for just a second. So every time in this class, so this won't be the last time you hear it, let me see if I can get you interested in kinases. If you thought you had a heart attack and you went to the hospital, and they're going to test you. They're gonna do some blood work, they're gonna do an EKG, which we're gonna do this lab, and we're gonna do a couple weeks in the lab, 3 weeks. So that you guys see your own heart beating, follow the waves, it's very cool. They might do an echocardiogram, they might send you up to the lab and do a stress test, they might, all kinds of things they can do, but they're gonna look in your blood for some things. So one of the things that you might go, okay, that kind of makes sense. They're gonna look for potassium ion concentrations. In your blood, they're gonna wanna know about that. And they're gonna wanna know specifically about the kinases that are specific for energy production in heart muscle. So here's what the kinases do. They're called CPKs, or creatine phosphokinases. The kinases in the heart take phosphate groups, add them to use energy to make new energy. So creatine phosphokinase adds kinases to ADP and you get more energy. Now, if you have a heart attack, your heart muscles know they need the energy and they overproduce the cardiac version of this kinase, and it'll be in your blood. The test, you have high CPK levels, they know likely you had a cardiac event. Now, you don't have to know that part, but I tell you that to try to get you interested, because this is what kinases do in the human body. When you hear that word from me, the first thing you should think is kinases are activators. They're activators. Things that are not working are made to work when the kinase acts. And the way kinases do that is to add phosphate groups. Isn't that weird? Life itself depends on the addition of phosphate groups to things that are not working. For example, adenosine diphosphate is a beautiful molecule, but it is of no use until you add a third phosphate to make adenosine triphosphate. Now you got something you can use, because the third bond is where all the energy is. In our body cells, when you add phosphate groups to proteins, like the cell has already been making some proteins in here that you need, and I'm going to give you one. It's already been making them in here, proteins that you need, but they're not working. So you can't use them. When the protein kinase is activated, that's what it does. It adds a phosphate group to the inactive protein. Now you can use it. So, as far as I know, every protein in the body, every protein that you use in human physiology needs to be phosphorylated in order to be active. And so that's what this second messenger does. It activates the kinase, the specific kinase that is going to trigger the cellular response by adding a phosphate group to something that's not working and make it work. Okay, so you want an example? Let me give you an example. So we're talking about adrenaline, right? The fight or flight response. Adrenaline is a hormone, and it's coming up in this presentation. I'll show you its structure, we'll talk about it again. But it's in physiology, we call it epinephrine, made by the adrenal gland. And when it binds to your heart, it makes your heart rate go up. So we're going to talk a lot more about that one. But I want to do this one with the sugar stuff, because I want to really talk about sugar a lot over the next couple of hours with you. Whenever you need energy, your body's going to elevate your sugar levels. And stress responses do this. Everybody knows that whenever people are under a lot of stress, they tend to gain weight. You guys notice this? And part of the reason for that is the stress hormones actually cause unnatural elevations in sugar levels. There are a number of adrenal hormones that do this. And my dad, for example, has been treated for the past 5 years now for prostate cancer. Very aggressive. They did a monoclonal experimental drug on him, and it appears to have worked. That's the answer to prayer. So grateful to still have my dad around, and it looks like he's beat it. And it's fascinating to me. But one of the things they've had to do over the course of time, because of the effect on the adrenal, is to give him adrenal corticotropic drugs to help the adrenal function. And even though he's taking low doses, it causes his sugar levels to be elevated. And when your body has elevated sugar levels, it will turn that sugar into fat and store it as energy in the form of lipid. Very cool process. He's not excited about it because for him, for the past 5 years, for him not to gain weight, he has to eat broccoli, sprouts, and celery for every meal. I mean, I'm over-exaggerating, but you follow me. Grass and leaves is all he can eat because he's already got elevated sugar levels, and even with that, he's gaining weight. So his cheeks are kind of pudgy, and he hates it. I keep telling him, Dad, you can't beat yourself up. Like, you beat the most aggressive prostatic cancer cells. It's okay that you're 20 pounds overweight. We're cool with that. Yeah? So sugar's a big deal. I'm just trying to keep you interested. Sugar's a big deal. And one of the ways that we elevate sugar is by using epinephrine, adrenaline, in your liver to cause the liver to release free sugar. Your liver is a storehouse for sugar in your body, and when you're stressed, epinephrine levels are high, they bind to receptors in the liver that cause sugar levels to go up. Cuts up the glycogen, releases the free sugar, and your sugar levels are going to go up. Your body is using the sugar for energy to deal with the stress. Okay, so here's where it connects. This mechanism is the mechanism that adrenaline uses in your liver to get sugar to be released. So here's what happens. The adrenaline binds to the receptor, it activates a G protein. The G protein activates adenylate cyclase that causes ATP to be converted to cyclic AMP that activates a family of kinases that phosphorylate enzymes that cut up glycogen. Did you get the connection? So inside the liver cell, there's stored sugar in a polymer. But you need a protein enzyme to come along and chop that polymer up to get the sugar to be released. And the way you do that is by activating a protein that's already in the liver cell. How do you activate it? By adding a phosphate group to it with a protein kinase because of the cyclic AMP levels. Did you get it? Okay, so I give you many more examples, but that one I think should resonate, and it will help us later as well. You can put this connection together. Now, in the laboratory, there's another second messenger system, and here it's got a phospholipase C, it's got a phosphonol triphosphate, and a diacylglycerol right here, and it runs down here to the ER and it releases calcium, and then calcium binds to another protein called calmodulin, and the calcium calmodulin activates another kinase. On Monday, how many of you guys remember seeing this? The phospholipase C, second messenger system. Calcium calmodulin should be yarn. Okay, I'm seeing that. Okay, so there's a whole other big thing to be told here, and we're not going to fight with all of that. The truth is, the adrenaline has receptors for both of those second messenger systems, which means when it hits the liver, it's a double-barreled sugar approach. Both alpha and beta adrenergics are there, and they use different second messengers with the same outcome. So very, very powerful hormones that are able to activate protein kinases. Okay, let's do one more here as an example to link me to the lipids, all right? I'll hold on. I think that's good for the water soluble. What questions do you guys have? This is always something students struggle with, the second messenger system stuff. You got a sense about what kind of hormones we're talking about here. So here we're talking about amino acid-based hormones, and Warren needs to name a bunch of those for you. So we're going to start in the brain coming up. And every time I name one, I'm going to say, y'all, look, this thing is made out of amino acids. From the fall, y'all remember talking about dopamine and serotonin? Those are neurotransmitters, yeah? And I told you about stuff they do, but they're built out of amino acids. So when the neurotransmitter is released and it does its thing, if that thing involves something produced by the cell, like a hormone, then how did it do that thing? This is how it did it, right here, using a second messenger system. Okay, good. So let's see if we can get some questions. Yes?
So after this, so the second messenger is what activates is what triggers the kinase to do its job.
Yep,
Correct?
that's right. Yeah, and so this is the critical step right here, because the hormone can't do anything unless the second messenger activates a kinase. And whatever second messenger system you're talking about, these steps will be the same. Ultimately, for water-soluble hormones, for hormones made from amino acids, you have to activate kinases in order for them to do their work. Yes?
The hormone is like essentially an instruction to create a certain type of protein, correct?
So in this case, Olivia, the hormone is probably a protein itself.
Okay.
The receptor's a protein. The G thing is a protein. The adenylate cyclase is a protein. Yeah? The kinase is a protein. So the whole thing is the story of protein interaction. But at the end of the day, inactive proteins in the interior of the cell get activated by the kinases. And the kinases are always activated by the second messengers. And there are a number of them. So cyclic AMP, diacylglycerol, phosphoinositol triphosphate, calcium, these are all second messengers that we know now. This is the one we know the most about. Yeah.
So whatever's phosphorylated is what causes the gland to release the
The
hormone.
cellular action. Yeah. Whatever it is I'm doing now. So the activity that happens as a result of the hormone is caused by the phosphorylation event.
And then after that, what comes that action is what releases.
So, yeah, so I mean, it depends on which hormone you're talking about.
Okay.
So for the one I'm talking about here right now, for the sugar stuff in the liver, the action then would be glucose being released out of the cell. Now, there are other parts of that as well. I mean, it just gets more and more cool as you go. Because the other thing that adrenaline causes is the activation of the channel proteins for the glucose itself. And that's also something that insulin does. So insulin uses a second messenger system, the receptor itself activates it, but then the end product is the phosphorylation of what we call a GLUT4 carrier protein. It's actually the protein that sits in there. So like if you don't bind insulin to the cell and activate the second messenger system, then you're never going to get the glucose to go into the cell to be stored, because it's responsible for putting the receptor, the channel for glucose, in the membrane. So the lack of insulin in the human body is catastrophic. Yeah.
Oh, I was gonna ask, is kinase, the protein kinase is the only protein that is phosphorylated?
The protein kinase is the phosphorylation initiator. it causes the phosphorylation event. And the phosphorylation event is the activation event for whatever protein you're talking about. So that's why I use the heart thing to try to get you to connect with that. The kinase there is adding a phosphate group to ADP. Now you have energy. A kinase did that. So when you go to the hospital, you're going to see, you know, in the blood list there's your CPK number right there. If that number's elevated, this is not good news. Yep.
So kinase, so basically kinase transfers ADP to ATP, basically, kind of?
It causes the addition of phosphate. So ADP is adenosine di. Adenosine, ATP is adenosine. How did you get the phosphate on there? Kinase did that. Kinase did that. Yeah. Good. Okay, everybody's got this one. All right, so this is the water soluble one. This one is the steroid one. So let's make the observation that we've pointed out a couple times here. This one shows you that the receptor protein is inside the cytoplasm. So here's the membrane right here, and here's the receptor protein right here. Now, this is not true of all steroids. Some of them have them in here. There's a chaperone system that you're going to go through in the lab, but I'm not going to ask you on the lecture to tell the whole chaperone story. I want you to get the gist of what's happening here. All right? So here comes the steroid hormone. Where did it come from? Two places. Where did the steroid hormone come from? Either the gonads or the adrenal cortex. That's right. So limited number of hormones here. So here we're talking about some version of testosterone, or some version of estrogen or progesterone, or we're talking about the one I'm going to show you, aldosterone, or the stress hormones, or the androgens. That's it. The list is limited for the steroids. So we're going to talk about all of them when I show you the adrenal gland, at least by way of outline. But here's the steroid hormone. Here it comes. Now, is it going to have problems going through the lipid bilayer?
No.
No, it's very nonpolar. It's going to go right through. When it gets through the lipid bilayer, it's going to bind to the receptor. Now, the receptor will be inside the cell somewhere. It'll be either in the cytoplasm or the nucleus, and that's good enough. That's good enough. The receptor will be inside the cell. All right. The next step is the hormone and receptor complex will bind directly to the DNA. So is this thing going to have a problem with the membrane of the nucleus? No. It's going to bind, and y'all, the details of this we know for every lipid hormone, every one of them. Like, what are the base pairs that the dimer of the receptor actually binds to? I mean, to hormone response element, there's a spacer in the middle. It forms a homodimer for all the steroids. For thyroid hormone, it forms a heterodimer with 9-cis-retinoic acid. I'm just spewing out information here. And you don't have to know. I'm just telling you, we know all of how these things work. The gist of it is the hormone receptor complex binds directly to the DNA. At that point, we call it a promoter or a transcript initiator. I see some of you nodding. You had a little microbiology. This came from your freshman biology class. Y'all heard about promoters and transcript initiators? I saw a couple of nods. Okay, so it's a promoter or it's a transcription initiator at this point. So transcription is standard science information here from high school and junior high. Transcription is going to result in the formation of a new protein. And so the binding of the hormone receptor to the DNA causes the formation of a messenger RNA in the nucleus. The messenger RNA then moves into the cytoplasm where it finds ribosomes, and at that point it gets translated. So the transcript in the cytoplasm gets translated into a new protein. What is the ultimate outcome of the binding of a steroid hormone to its receptor? A new protein. What is the ultimate outcome of a water-soluble hormone to its receptor?
To activate a
An
protein.
activation of an already existing protein. That's the difference. Now let's do an example for this one like we did with the water-soluble. Yeah, we're doing great. Okay, so let's do an example of this one. So one of the hormones we're going to talk about quite a bit over the next couple of hours together is aldosterone. A-L-D-O-S, it's not written on here. A-L-D-O-S-T-E-R-O-N-E. Now, I'm going to skip ahead on the slides and show you the picture, but please don't panic yet. I'm going to go through it stepwise with you. I just want you to see it right now, just see a picture of it. So many cool things to talk about in the hormones. So many cool things. All right. So this is the picture that I'm going to use later to talk about aldosterone. So you can see its thing here in the middle. And when I look down here, if I take my eyes from this piece of the picture, aldosterone coming out of the adrenal cortex down to here, what's the ultimate outcome of aldosterone? Yeah, elevated blood pressure.
Thank you.
All right, so whatever this is, whatever this aldosterone control thing is, we're going to talk about it in terms of blood pressure. So it's really important. It's part of a big system called the RAS, R-A-A-S, the renin-angiotensin-aldosterone system, and it is the hormone way that we control blood pressure. And so drugs designed to impact blood pressure are going to get plugged into this pathway that I'm going to teach you. Okay, so this is why I'm using this as an example for aldosterone. So the first thing you know is that aldosterone, here we go. The first thing we know is that aldosterone is made by the adrenal cortex and it is a lipid. So this mechanism is in place. And so the question then should be, what is the new protein? Like for the sugar story, what ultimately got phosphorylated to make sugar levels go up? Enzymes that chop up glycogen. Okay? This one is going to make blood pressure go up right here, whatever this protein is. And I picked it on purpose because you've heard me talk about it. Do you guys remember the nervous system talking about sodium potassium pumps? Somebody tell me what the role of sodium potassium pumps are in the nervous system. Somebody want to take that one?
Action potential sequence.
Say again?
Action potential sequence.
Okay, so they have to do something so that you can make action potentials and have signals going down neurons. What exactly do those proteins do in the nervous system? We call them sodium potassium pumps for a reason. What do they do?
They move sodium and potassium.
You say? Go ahead, say.
They move sodium and potassium.
Yes. How?
Down the axon
Does
turn?
So y'all are hitting on it. It makes me so happy. So sodium is transported out, and potassium is transported in. So this makes the gradient that the neuron is going to use to send the action potential. Do you remember that? Okay. If I'm in the kidney and I want to regulate how much salt and water I'm getting rid of or keeping, this is the protein that dictates it. So in the kidneys, we're not interested in making action potentials in the kidney tubes. we're trying to regulate how much salt and water is in the blood. But the kidneys use the same protein to do it. So in the kidney tubes, like where you're making urine, aldosterone binds there, causing those tube cells to make more sodium potassium pumps. When that happens, it causes the kidney to move sodium out of the urine and back into your body. As the sodium gets pumped out, chloride follows it. Chloride's just the buddy that wants to be with sodium. And that sodium chloride, don't know what that is? That's salt. And so the addition of sodium potassium pumps causes kidneys to move salt out of the urine that's being produced and put it back in your body. And so if the salt is kept, what will the water do? Water follows the salt. And so the ultimate end game here is salt retention causes water retention, causes blood pressure to
Rise.
That's how aldosterone does its thing. So you heard me say this in the fall, and it's true. Like, what is the most important protein in human physiology? What is the number one reason why you make energy for life? To operate sodium potassium pumps. Because you're not going to be able to send action potentials to make muscles contract or anything else without them. You're not going to be able to do regulation for blood pressure, blood volume. And I have more coming this semester for heart function. Okay, so that's why I use it, yeah? What's the new protein made here? Aldosterone binds to its receptor. The receptor with aldosterone binds to the DNA. The messenger RNA is formed and gets translated in the cytoplasm into a new protein. And that protein, for your example, is aldosterone. What's the impact of aldosterone then in this cell? Sorry, let me say it again. I said that wrong. Aldosterone binding mRNA translated into a new protein. The new protein is not aldosterone. What's the new protein? The sodium potassium pump. And what's the role of that new protein, that sodium potassium pump?
Regulate salt.
Salt retention in the kidney.
Does that protein have a name for the sodium potassium pump?
No, that's its name.
That is-
The sodium potassium ATPase pump. That's the name of it. Yeah. Yeah.
Is that true for all?
It attaches to the receptor.
So every steroid hormone is going to cause the formation of some new protein.
No, I mean like for the sodium potassium pump. Like, is our body continuously producing that protein?
Absolutely, it's happening right now.
It understands, so it makes it into
Yeah.
a messenger
Okay,
RNA.
so let me introduce you to what we're going to do on Friday here. Perfect. And then I'll let you go. So on Friday, we're going to begin our discussion about the master hormone control system. This system is the connection between the hypothalamus of the brain and the pituitary gland. So any endocrine discussion about hormones should start here. Notice where I am. This is the sagittal section of the brain right here. So I've been cut right there and here. So here's my cerebral hemisphere. You guys remember from the lab test? Corpus callosum, lateral ventricle, thalamus, hypothalamus, pituitary connected to the bottom. Yeah, that was on your lab final. Okay, so this is the hypothalamus blown up, and you can see that the hypothalamus communicates with the pituitary in two primary ways. One, there are neurons in the hypothalamus that actually make hormones that are released by the posterior pituitary. So we'll do this one first on Friday. The second one is there are neurons in the hypothalamus that produce hormones that are transported to the anterior pituitary by way of the circulatory system. Now, collectively, the hormones that are produced by the hypothalamus released by the posterior pituitary, and the hormones produced by the hypothalamus that are transported by way of blood supply to the anterior pituitary, collectively, those two groups of hormones, I would defy you to find me a hormone that is not either directly or indirectly impacted by these hormones. So they're really important. So this is what we'll do on Friday. I'm going to teach you these two, and then I'm going to teach you all the hormones that are part of this system right here. So we'll have some new terminology to get all these guys out. After I've done that, then we're going to do a walkthrough. We're just going to hit glands as we're walking through. Just so you know, I'll say this on Friday as class ends, but next week, Monday's a holiday, so Monday lab can't be, and I don't want y'all to get off sequence, but I'm going to make an assignment, a lab assignment, that both the Monday and Wednesday labs will do on your own time. So I won't require the Wednesday lab to come to lab. I'll be sitting in there if y'all want to come talk, but it's a little 24-minute video that's going to do what I'm doing in class. That way, you'll hear it over the weekend, you'll come to class on Wednesday, and there's more. I'm spewing out the same information. All right?
What does it do?
It's going to be due next week, so Wednesday, Friday next week. Okay, I'll see you Wednesday in room 214 at 12:30.